Students, today we will learn about transgenic animals. So let's begin. The dependence of man on animals such as cattle, sheep, poultry, pig, and fish for various purposes like milk, meat, egg, wool, etc. is well known. Improvement in the genetic characteristics of livestock and other domestic animals, example, high milk yield, weight gain, etc., in the early days was carried out by selective breeding methods. This technique primarily involves a combination of mating and selection of animals with improved genetic traits. Although selective breeding is very time consuming and costly, it was the only method available till some years ago to enhance the genetic characteristics of animals for larger animals with long gestation period. It might take several decades to create a desired character by conventional breeding. With the advent of modern biotechnology, it is now possible to carry out manipulations at the genetic level to get the desired characteristics in animals. Transgenesis refers to the phenomenon of introduction of exogenous DNA into the genome to create and maintain a stable heritable character. The foreign DNA that is introduced is called transgene, and the animal transgenes is said to be transgenic. The transgenes behave like other genes present in the animal's genome and are passed on the offsprings. Thus, transgenic animals are genetically engineered or genetically modified organisms with a new heritable character. It was in 1980s the genetic manipulation of animals by introducing genes into fertilized eggs became a reality. Now let's know about the importance of genetic transgenic animals in general. Transgenesis is now become a powerful tool for studying the gene expression and developmental processes in higher organisms. Besides the improvement in their genetic characteristics, transgenic animals serve as good models for the understanding the human diseases. Further, several proteins produced by transgenic animals are important for medical and pharmaceutical applications. Thus, the transgenic farm animals are a part of the lucrative worldwide biotechnology industry with great benefits to mankind. Transgenesis is important for the improvement of the quality and quantity of milk, meat, eggs, and wool production, besides creating drug resistant animals. Now, let's know about milk as the medium of protein production. Milk is the secretion of mammary glands that can be collected frequently without causing any harm to the animal. Thus, milk from the transgenic animals can serve as a good and authenticated source of human proteins for a wide range of applications. Another advantage with milk is that it contains only a few proteins, that is casein, lactalbumin, immunoglobulin, etc. in the native state. Therefore, isolation and purification of a new protein from milk is easy. Now let's know about commonly used animals for transgenesis. The first animals used for transgenesis was the mouse. The super mouse was created by inserting a rat gene for growth hormone into the mouse genome. The offspring was much larger than the parents. Super mouse attracted a lot of public manipulation. Mouse continues to be an animal of choice for most transgenic experiments. The other animals used for transgenesis include rat, rabbit, pig, cow, goat, sheep, and fish. Now let's know about transgenic mice. Mouse is the animal of choice for transgenic experiments. Being a small animal, being a small animal, it can be easily handled, and mouse is regarded as researchers friendly by biotechnologists. Another important reason for of choosing mice for transgenic experiments is that it produces more eggs. Normal mouse is 5 to 10 eggs. Superovulated mouse can yield up to 40 eggs. Unlike the large domestic animals, in up to in the last 2 decades, hundreds of different genes have been introduced into the various mouse strains. And as such, the methodology of transgenesis is well developed with laboratory mouse. Transgenic mice have significantly contributed to the understanding of molecular biology, genetics, immunology, and cancer, besides creating animal model for several human genetic diseases. There are three methods for introducing a foreign gene into mice, and in fact, the same method are applicable to other animals as well. First is retroviral vector method, second is microinjection method, third is embryonic stem cell method. It may be noted that the term transfection is used for introducing a foreign gene into animals. This is quite comparable to transformation that is carried out in lower organisms. Now let's know about retroviral vector method. The transfer of small pieces, that is 8 kilobase pair of DNA, can be effectively carried out by retroviruses. This method, however, is unsuitable for transfer of larger genes. Further, even for small genes, there is a risk of losing some regulatory sequences. Above all, the biggest drawback is the risk of retroviral contamination in the products, particularly in foods for human consumption obtained from transgenic animals. Because of these limitations, the retroviral vector methods is not in regular use for transgenesis. Now let's know about microinjection method. The introduction of DNA by microinjection method involves some steps. Let's know about these steps. First point is the introduction. First point is the young virgin female mice, that is 4 to 5 weeks age, are subjected to superovulation. This is achieved by administration of follicle stimulating hormone, that is pregnant mare's serum, followed by 2 days later human chorionic gonadotropin. The superovulated mouse produces 30 to 35 eggs instead of normal 5 to 5 to 10 eggs. Second is the this female mice are mated with males and sacrificed on the following days. The fertilized eggs are removed from the fallopian tubes. Now, third point, the micro manipulation using a microinjection needle and the holding pipette. The DNA is injected into the male pronucleus of the fertilized egg. Adequate care must be taken to ensure that while the elastic nuclear membrane is punctured, the needle does not touch the nucleoli. A dissection, the dissection microscope can be used for identifying the male pronucleus and for the microinjection. Fourth point is the eggs with the transgenes are kept overnight in an incubator to develop to a two-cell stage. These eggs are then implanted microsurgically into a foster mother, that is pseudo mouse, pregnant female mouse, which has been mated the previous mite with vasectomized or infertile male. The foster mother can deliver POPs after 3 weeks of implantation. The presence of transgene in the POPs can be identified by polymerase chain reaction or southern blot hybridization. The mouse carrying the foreign gene is the transgenic founder from which pure transgenic lines can be established. The microinjection method involves several steps and none of them is 100% for the 100% efficient for any animal to develop into transgenic animal. In case of mouse, it was found that 65% of the fertilized eggs survive microinjection procedure. About 25% of the implanted eggs develop into pups, and only 25% of them are transgenic. Thus, if one starts with 1000 fertilized eggs, only 30 to 50 transgenic pups may be produced. That is, 3 to 5% of the inoculated eggs develop into transgenic animals. Now let's know about the limitations of microinjection method. Besides the low efficiency, there are some other disadvantages in this technique. The foreign DNA randomly integrates into the host genome. Sometimes even many pieces of DNA get incorporated at a single site. Further, transgenes may not be expressed at all, or sometimes underexpressed or even overexpressed. All these processes will disturb the normal physiology of the transgenic animal. In addition, microinjection procedures are time consuming, costly, and labor intensive. Despite all these limitations, this technique is routinely used for the production of transgenic animals. Now let's know about embryonic stem cell method. Cells from the inner cell mass of the blastocyte stage of a developing mouse embryo can proliferate in the cell culture. These cells refers to as pluripotent embryonic stem cells are capable of differentiating into the other types of cells including germ-line cells when transferred to another blastocyte embryo. The embryonic stem cell technology basically involves the introduction of a foreign DNA into a embryonic stem cell. Embryonic stem cells in culture can be subjected to genetic manipulations without changing their pluripotency. Foreign DNA can be introduced into embryonic stem cells by electroporation or microinjection. The desired genetically engineered cells with transgene can be identified by selection procedure using a marker gene or PCR analysis. The transfected cells can be cultured, introduced by microinjection into blastocyte and then implanted into foster mother, that is pseudo pregnant female mouse. By this way transgenic founder mice are produced. Transgenic lines can be established by suitable breeding strategies of the founder mice. Now let's know about selection of transgene containing cells. Several strategies have been developed for the selection of transgene containing cells. The important ones now we will discuss. Selection by using a use of marker gene coding for thymidine kinase. Let's know about this. It is worthwhile to know the role of thymidine kinase to understand its utility as a marker gene. There are two pathways for the synthesis of deoxyribonucleotides, that is DATP, DGTP, DCTP, DTTP, the basic units of DNA structure. One of the salvage pathway that recycles the degraded nitrogenous bases formed from DNA. The other alternate pathway is an endogenous synthetic pathway from different precursors, that is glycine, aspartate, glutamine, methyl tetrahydrofolate, etc. The enzyme thymidine kinase, in short TK, is involved in the salvage pathway. TK phosphorylates thymidine to produce thymidine monophosphate, in short DTMP, which is finally converted to thymidine triphosphate DTTP. The gene that encodes the enzyme thymidine kinase can be used as a marker to determine whether the transgene has been inserted. The mammalian cells are capable of synthesizing DATP by salvage pathway and endogenous synthetic pathway. The cells lacking TK gene cannot produce DTTP. If such cells are cultured in a hat medium containing hypoxanthine, that means H, aminopterin, that is A, thymidine, that is T, they cannot grow and therefore die. This is because thymidine cannot be utilized in the salvage pathway due to lack of enzyme thymidine kinase. Further, aminopterin blocks the endogenous pathway by inhibiting the enzyme dihydrofolate reductase required for 1-carbon metabolism. If a transgene is joined to a TK gene inserted into a mammalian cell, and then the cells can grow in hat medium. This is possible only if the TK gene is incorporated into the mammalian cells, and logically the cells that survive in hat medium carry the transgene. In this fashion, thymidine kinase can be effectively used as a marker gene. There are other enzymes that serve as markers for identifying transgene insertion. These include dihydrofolate reductase resistant to methotrexate and neomycin phosphotransferase resistant to antibiotic G418, and PCR analysis for selecting transgene containing cells. The last one is a more direct and recent method and is successfully used for detecting transgene containing cells. Now let's know about promoter sequence to facilitate transgenesis. In the early experiment on transgenesis, the mouse metallothionein, in short MMT, gene promoter was used. MMT gene encodes for a metal binding protein that is involved in metal homeostasis. The foreign gene, that is a rat growth hormone gene, can be linked to a promoter sequence of MMT. By doing so, the promoter switches on the growth hormone gene when the MMT is activated by a metal in the environment. Example, cadmium. Thus, the metal inducer, in short CD, can stimulate the promoter to facilitate the transgene, that is growth hormone gene, to express. Therefore, the addition of cadmium triggers the growth hormone production. Now, let's know about gene knockout. By inserting a transgene, foreign gene, into a chromosome, a new function is introduced while producing transgenic animals. On the other hand, in the process referred to as gene knockout, an existing function can be blocked by destroying a specific gene. The target gene disruption can be carried out by the incorporating a DNA sequence, usually a selectable marker gene, into the coding region. Selectable marker gene, we can say it as in short SMG. The chromosome carrying the target gene with 4 exons with flanking sequences is subjected to homologous recombination with a vector carrying selectable marker gene. The homologous recombination results in gene knockout, that is disruption of the target gene. In the gene knockout, the loss of function occurs in transgenic animals. This is in contrast to gain of function that takes place by introducing a foreign gene. Now let's know about application of gene knockout. Disruption of the target genes. Gene knockout is important for understanding the development and physiological consequences of in an organism. Further, the biochemical and pathological basis of several human diseases can be appropriately understood by inactivating specific genes. About 20 about 200 knockout mice have been so far created to serve as animal models for the study of a large number of human abnormalities and disorders. In fact, knockout mouse which lack genes for a single organ or a organ system can be produced. And now let's know about each artificial chromosome in transgenesis. In the conventional transgenesis, small-sized transgenes, that is that is greater than 20 kilobase pair, are transferred through vectors. This involves a risk of losing some important sequences, including the regulatory ones. Further, several genes exist as complexes and are too large to be handled by conventional vectors. Each artificial chromosomes can carry large-sized genes, that is, size range from 100 to 1000 kilobase pair, and are effectively used in transgenesis. By using microinjection technique or transfection of embryonic stem cells with YAC, each artificial chromosomes transgenic mice have been produced